Okay. Enzyme activity. So in this session, we will look at what are some of the conditions that would change enzyme activity. But this animation shows again the specificity of enzyme activity, enzyme catalyzed reactions. So if the substrate doesn't fit the active site, then the enzyme just wouldn't work. Okay, so enzyme activity is monitored by the rate of the conversion of substrate into the product. So basically the rate of reaction, right? Remember how we measure the rate of reaction? We measure the time it takes for a reaction to basically happen, right? So a lot of times we actually use 1 over time, so the reciprocal of time, to measure the rate. So basically the higher the time it takes, the smaller the rate of the reaction. Okay, so any conditions that affect frequency of interaction between substrate and enzyme affect enzyme activity. So there are 3 major factors: pH, temperature, and concentration of substrate. Concentration of substrate is relatively easy to understand because we learned that from chapter 7. The rate of reaction will be dependent on the concentration of reactants, right? The substrate is our reactant. So higher concentration of substrate, of course, will result in higher rate of reaction. So that actually doesn't have much to do with enzyme. But pH and temperature will really very specifically affect enzyme, the structure of enzyme, and therefore enzyme activity. Let's have a look. So since enzymes are mostly proteins, protein has this process called denaturation. So what will cause denaturation? Well, extreme pH and temperature, or temperature actually. So what they do is that they destroy the tertiary or quaternary structure of the protein. Therefore, the protein permanently changes its shape. Okay. Once you change the shape, you change the shape of the active site. Then that protein is no longer useful and basically no longer capable of being bind to a substrate. As a result, the enzyme activity, or the protein basically, is useless. Or we can say they die, right? But in the process of denaturation, the primary structure, basically the covalently bonded amino acids, is intact. So extreme temperature and pH, they don't really destroy the sequence of amino acids that's in the polypeptide chain, but they do change the shape of that protein, sometimes completely destroy the shape of protein, and therefore render them basically useless. So that's denaturation. Okay. So remember that we talk about that Switter ion, and like as an amino acid, they have amine groups on amino acids that are capable to accept protons, and then they have carboxyl groups that are acid and are able to donate protons, right? So in a neutral solution, any amino acid, if they are hanging around by themselves in a neutral solution, then they will turn into a Switter ion with a NH3 positive end and a COO negative end, right? So that's a very common structure. Like once a protein or amino acid is actually in neutral solution, it will turn into that, the whole polypeptide chain will turn into that Switter ion, right? But what happens when amino and carboxyl functional groups on the side chain in the solution, right? They will behave exactly the same way. So if you have a side chain that has an NH2 group, in solution, it will naturally become NH3 with a positive charge, right? Or if you have a COO, so basically a Cu group on your side chain, also in a neutral solution, it's going to donate its hydrogen. Yeah? So they will behave the same way. And because they behave that way, they naturally have charge in the neutral solution, right? Once they have charge, they are able to form that ionic interaction that are responsible for the tertiary structure of a protein. Okay, so when we talk about ionic interactions to form that tertiary structure, mostly we're actually talking about the ionic charges that actually form by NH3 and COO on the side chain. So these two groups can appear in separate side chains, and when they appear, they have the positive and negative charges, and then they can form that tertiary structure. So they're actually responsible for part of the tertiary structure. However, what's going to happen if you change the pH, let's say, of the solution? What's going to happen? We know that for sweeter ion, a change in pH will, for example, if the pH becomes lower than it should be, then the COO negative will become a real cool at low pH, right? Or if the pH becomes weirdly high, then the NH3 group will lose its hydrogen to go back to become NH2 group at high pH. So we talk about when it's sweeter ion will change its charge, become cation or anion in different pH. But now we're talking about the Cu and the NH3 on the side chain. They will also change as pH changes. And if you can see here, these are two polypeptide chains. Okay, so these two chains are bonded together because of the ionic interaction between the Cu and the NH3. Right? That we can imagine that is a protein in its natural environment, in the environment where it should be when it's functioning properly, right? But if you change the pH, then if the pH goes low, then the Cu loses its charge, disrupts the ionic interaction. If the pH gets high, the NH3 loses its charge and disrupts the ionic interaction. So you can see that the changes in pH alters the charge of the side chains containing the amino or carboxyl functional groups. As a result, the tertiary structure is disrupted. So that's how pH changes enzyme action. Like basically it changes the charges of those ionic interactions and therefore destroys the tertiary structure of a protein, and therefore the enzyme is no longer useful. Temperature. How does temperature change the enzyme activity? Well, enzyme function is impaired whenever it's above or below the optimum temperature. So it's the same thing. With pH, enzymes actually works in a very narrow range of pH that it's suitable for it. Like if the pH changes, the tertiary structure destroyed, and then the enzyme is no longer working. They also work in a really narrow range of temperature. Because if the temperature goes extremely high, you increase the kinetic energy of the molecules, right? That will disrupt the structure of enzyme by breaking the intermolecular forces that's responsible for the tertiary or quaternary structure. Remember that the three intermolecular forces, dispersion forces, dipole-dipole, and hydrogen bond, they are really susceptible to temperature change, right? The whole reason that we have something called boiling point is because temperature changes the intermolecular forces and therefore changes the state of a substance by turning it from liquid to gaseous. The same thing happens here. The tertiary structure are like the intermolecular forces are actually responsible for the tertiary or quaternary structure of a protein. But when the temperature increases, the intermolecular force gets destroyed, and as a result, the active site of the enzymes are no longer effectively catalyzing the reaction because the shape has changed. The intermolecular forces gets weakened, and the shape of the enzyme has changed. The shape of the active site has been changed, and therefore the active site is no longer active. Okay, so this is what happens with high temperature. So high temperature essentially destroys the protein, just like how you change the pH also destroy the protein. Okay, that's also why when you're sick you lose appetite, because especially when you're sick with a fever, the enzymes in your digestive system literally die because of the high temperature. They get denatured. So those enzymes die, and therefore you don't really have enzymes to catalyze the food that you're about to eat. And as a result, your body actually adjusts by not letting you eat any food. So it takes your appetite away. Okay? So what happens when temperature is too low? Well, low temperature doesn't really kill a protein, right? Low temperature does not destroy the tertiary or quaternary structure of the protein, but it does decrease the kinetic energy of basically your substrate as well as your protein molecules, right? Your enzyme molecule. So they actually result in less frequent and less energetic collisions between the molecules. So when temperature is too low, enzyme activity is low as well, but that's not because the enzymes are destroyed, but more really because of a rate of reaction problem. That is, low temperature, low kinetic energy, so low frequency of collision and low energy in collisions between the molecules. That resulted in low rate of reaction and manifests in low enzyme activity. Okay, so if we plot a graph, we can see that for basically almost every enzyme, we have a graph with reaction rate versus time. You can notice that as long as it's around the range of optimum temperature, enzyme activity is high, as reflected in rate of reaction is high. When temperature goes low, the enzyme doesn't die. You will always have enzyme activity, so it doesn't go to zero, but the reactivity actually decreases. Enzyme activity decreases. But when temperature gets high, you notice the line also goes down. And when temperature goes high, the line actually can touch zero because there will be a temperature that you kill all the protein that you're investigating. Okay, so low temperature doesn't go to zero, but high temperature does go to zero. And the best is the optimum temperature, somewhere in the middle. Especially for the enzymes in your body, they normally functions around 37 degrees. So go back to look at pH. So pH, like enzyme, operates effectively only within a narrow range of pH, right? The pH at which the enzyme activity is greatest is known as the optimum pH. Because the intermolecular bonding between the side groups on amino acid is important for maintaining the structure integrity, the tertiary structure. So pH changes affects those groups, the behavior of those groups, and therefore affects the bonding. So extreme pH can cause denaturation of the enzyme and therefore render the enzyme basically useless. Right? So different enzymes at different parts of the body will have different optimum pH range, right? So for example, pepsin works in your stomach, so its optimum pH is around 2. Okay? Salivary amylase, that's basically the amylase in your saliva. That's why we always say that eat your food slower and chew more because it gives time for the enzyme to actually react, right? Just imagine if you swallow things really fast, the enzyme amylase goes with the food. That's okay. But the problem is that it goes with your food all the way into your stomach. And in your stomach, the pH is around 1 or 2. So amylase will not be effective there. That's why you got to chew more and allow enzyme to actually react in your mouth before you actually swallow it. Lastly, let's look at a saturation of enzyme activity. So first of all, when enzyme concentration increases, the rate of reaction obviously increases with it because, well, more enzymes, then more catalysts, then the reaction goes faster, and it increases infinitely as the enzyme concentration increases. The problem is that sometimes the enzyme concentration remains the same, but you increase the substrate concentration. Okay, this will have an effect where the rate of reaction will reach a saturation point. Initially, when you increase substrate concentration, the rate of reaction does increase because you always have free enzymes there ready to catalyze the breaking down of the substrate, right? But once it reaches a point where all the enzymes in your body are already occupied, then at that time, even if you increase the substrate concentration, the rate of reaction will not change because you're running out of enzyme. So this is what happens when you overeat. Some people feel like, oh, I'm in a food coma. Why are you in a food coma? Because you eat too much and all your enzymes in your stomach are already occupied. So you feel like this sense of indigestion in your body. And a lot of time, the medication you take for indigestion actually basically is supplying you with those extra enzymes that will work in your stomach that helps to break down the food. Okay? So when substrate concentration keeps increasing, there will be a point where the rate of reaction increases to the maximum. That's when all the enzymes are already working and no one is full.